Hello friends, good afternoon and welcome to EduSite live lectures. Dear friends, today in Introduction to Proteins we will be talking about protein misfolding and disease. To discuss this topic we have with us our subject expert Dr. Unnati Gulati. Dr. Gulati is Academic Coordinator at Consortium for Educational Communications, New Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Welcome dear students. We have been discussing in this lecture series about protein folding, and we have seen various thermodynamic and chemical aspects of how proteins get their tertiary structure or their functional structure. We will now on look at what happens to the protein if there is no tertiary structure or there is no native structure. So, we will be looking at what is the loss of this structure and what are its implications. You may understand proteins being the enzymes, and just with one example we can understand if an enzyme is not functional, if it is unable to recognize its substrate and hence is not able to catalyze a reaction, that means the whole metabolic pathway gets haywired. Simultaneously, if multiple such enzymes stop functioning, then there can be no biochemical pathways. Similarly, this can be applied to several processes: signaling, cell cycle, apoptosis. So, we will see that if there is any loss of function or other misfolding or aggregation that occurs with the protein during its folding, so what are those implications and what are the diseases that can be caused by them. So, today's lecture, it's our 9th lecture in the series, and we will talk about misfolding and protein aggregation. What is the thermodynamic basis? We will touch upon that. What are the structural changes there? What are the cellular consequences when protein aggregates? And what are the diseases that can be caused by protein misfolding and aggregation? So, we are familiar with this folding funnel, and we know that an unfolded protein has several conformations, and as it goes down this folding pathway, folding funnel, it encounters several local minima. These local minima are those contacts which eventually sum up to the native contacts, but many a times these contacts may be non-native also, that is, they may not be the perfect contact to achieve the native state. And accumulation of such non-native contacts over the time when it is following the folding pathway can result into an entirely different conformation of the protein as it goes down. So, you can see at the bottom of this folding funnel, I am showing here two minimas. One is the minima of the native state, and closer to it thermodynamically is another minima which is maybe an aggregated state. We will look at it very clearly in subsequent diagrams. So, when we are talking about folding funnel, we are looking at maybe one unfolded protein molecule and how it traverses through several conformations and reaches its native state. But is that the scenario when the newly formed protein polypeptide is emerging out of the ribosome? When the protein emerges and is released into the cytosol, many a times it is encountering a very dense, a crowded environment. This crowded environment, due to the concentration of several other proteins, other carbohydrates, lipids and small molecules present in the cytosol, causes high density of the cytosolic solvent system, and in that case all our water hydrogen bonding may be not as ideal as we had discussed previously when we were discussing all the forces, all the weak forces that were important for protein to attain its tertiary structure. So, in a crowded environment things are not as ideal, and only hydrogen bonding of the water is not what you are looking at because the solvent is so crowded. Also, when the protein is formed, it may be formed as a nascent chain, and there are several such molecules present in the environment, and the chances of them aggregating and not forming the ideal native state are very high. So, this is a diagrammatic picture of how crowded actually is the environment, and these dumbbell shaped organelles that you are seeing are the proteosomes, and we will also discuss what these proteosomes do in our next lecture. So, it is a fairly crowded environment, and in this crowded environment imagine a peptide which is a polypeptide having no native conformation starts getting small steps towards native conformation, and it may not be able to reach its native state. So, what is it that makes a native state different from an aggregated state? You have seen in the previous funnel model also that there is not much difference, but actually during the aggregation the free energy change depends largely on the concentration of the monomer, that is the monomer of the polypeptide which is just recently formed and released into the cytosol. At low concentration the monomeric state is very much favored, but as you see more and more crowding, as you see more and more high concentrations of this monomer, you will see aggregation will tend to occur. And there is a large barrier between an aggregated state and a folded soluble state. I would remind you here that not all aggregates are insoluble, we will talk about them also, but there is a distinct difference between an aggregated state and a soluble state, and that energy barrier prevents the free movement across the equilibrium. You cannot go very easily across from the native to the aggregated state. If you remember, we had spoken about the equilibrium that exists between the native and the unfolded state, and we said that is a very this equilibrium is very fine balanced. You will never find entirely unfolded or entirely native protein. So, that is the equilibrium we are talking about, but here when we are talking about native and aggregates, this equilibrium shifts very much if to the side of the aggregates, then it is very difficult for it to come back towards the native state. So, this is the two funnels that we will see. What happens when there is accumulation of aggregates or when there is misfolding occurring. So, looking at the first funnel, you will see that it is normal how we usually see our protein which follows this funnel and attains the native state from an unfolded state, emerging with more and more native like contacts and getting to the local minima which is the native state. And how separated it is from the aggregation funnel, and you can see this and this local minima of aggregation and folding are of equivalent energy, and to go against this barrier and reach the folding funnel, it is very difficult. So, this is the case of aggregation, and aggregation is a process which sometimes occurs autocatalytically such that even a small aggregate, if it is formed which is having only few polypeptides, can catalyze aggregation into longer filaments by accumulation of more and more such oligopeptides. So, you look at the diagram in the second in this right pane, you have the folding funnel, and subsequently you have aggregation where oligomers are getting formed by addition of more and more non-native like contacts, and you eventually end up in a fibril. So, here I am just talking about how folding funnel, which was in the case of a globular protein, is giving rise to some fibril like structure. Let us look at what is this fibril like structure, what are its characteristics in terms of its secondary structures, do they have any structure. So, we will look at those aspects. So, we can say that there is two aspects we were talking about, that is misfolding and misassembly. We can have a misfolding of a protein where it is a soluble protein which does not acquires the native like contacts and it becomes misfolded. Generally upon misfolding, the hydrophobic residues get exposed to the water towards the solvent side, and once they are exposed towards the solvent side, hydrophobic residues of one polypeptide chain may interact with the hydrophobic residues of the other polypeptide chain, coming closer and forming aggregates. So, this is the case of misfolding, And when such aggregates form, and they may result into a quaternary structure which is not functional, or they may result into fibrillar structures which comes under the misassembly. Eventually misfolding may result into misassembly because of exposure of the hydrophobic residues. So, you will encounter that when the proteins follow a folding pathway and they get into a wrong folding pathway, or they get entrapped into a local minima state, it is the misfolding. And these kind of diseases you will encounter when there is polyglutamine kind of motif present in a protein which causes it to aggregate. We will see more reasons and more implications of what causes the protein to become misfolded. So, misfolding diseases we will see are going to be based on misfolding, that is loss of function. We can have misfolding that leads the protein to become to not reach the place where it has to go and get incorporated and perform its function. So, there can be problems with the trafficking. What is trafficking? Trafficking is once the protein is in the endoplasmic reticulum, it releases out through either vesicles, and it gets to and travels through the vesicles to the respective organelles, maybe the mitochondria, chloroplast, where it finally gets introduced into the organelle and it has to perform its function. You must remember that not all the proteins present in these organelles, chloroplast and mitochondria, are from the DNA of the mitochondria and chloroplast. Many of them have subunits that come from the cytosol, or they are directed from the nucleus. So, trafficking is a major issue here. Also, many of the misfolded proteins can result in aggregates, and these aggregates could be loss of function, that is there is depletion of the enzyme where it has to be, or there is a gain of a toxic function, that is once the aggregate is formed, it may have a toxic function, or these toxic aggregates may accumulate within the cell and may disrupt the cellular functioning. So, there are going to be accumulations, loss of function, toxic gains of functions, and functional insufficiency, where you do not have appropriate or optimal level of enzyme to complete the metabolic pathway. And this can result due to stress responses, overwhelming of the chaperone system, where the proteasome is not working optimally to take care of all the misfolded and aggregated proteins, or there is a sequestration of the important transcription factors. There is an aggregate which is causing a pore formation within the cell wall. There can be excess of calcium that can result in this kind of aggregates. Oxidative stress, glutamate overload, as in the case of Huntington we have seen polyQ, we will see also, and mitochondrial dysfunction due to loss of function because there was improper trafficking of the protein. So, the misfolding of the protein is leading to 4 important inhibitions or 4 important implications. First is the gain of toxicity, where structural alterations that establish novel toxic functions, and these are we can think about it as a misfolded protein when gets aggregated, it should not cause any problem, but when all this junk gets accumulated in the cell, it causes lot of overload. Many a times all these proteins must be having some important relevant function into the cell, and if they are only going towards the aggregated equilibrium and not forming the native protein, then there is no protein to cause the normal functioning of the cell. So, there is both dual toxicity because of loss of function, insufficiency of the protein, and then there is gain of toxicity because of accumulation of the junk DNA. Also, there is mislocalization, that is to say in the organelle the protein never reaches where it has to incorporate or perform a particular function, and this is what you will see in the disease of cystic fibrosis. And accumulation, protein aggregates will form fibrils and various type of deposits. We will see those things, and then there is improper degradation. We will discuss in detail what is improper degradation. When I am saying degradation, that means there must exist a process or mechanism in the cell to take care of all the junk proteins which do not have any function or may have outlived their function. They have become old after serving their purpose, and they eventually go towards the pathway where they should be degraded and all the amino acids should be retrieved from them. So, there is a degradation machinery present, which is the ubiquitination, proteasome pathway and lysosome pathway, which we will discuss. So, if that machinery undergoes an overload, or that machinery undergoes some mutation that it is not able to do the clearance, then also accumulation will cause certain diseases. We have discussed in our previous lecture that protein misfolded is prevented by several chaperones, but this mechanism may not always be able to sustain the excessive misfolding or misassembly that occurs. So, I would like to remind you that chaperones were first discovered by Ron Lasky in the year 1978, and he found that there are some nucleoplasmin proteins which have a tendency to bind to the histones and cause binding of the DNA, and this was discovered in the amphibian eggs, and he thought that these are chaperones, but he did not apply this concept to a larger or broader term of proteins that can help other proteins to fold or assemble. And this idea was given by John Ellis, who gave the term chaperone in this paper in 1987, and the term chaperonin was given by Sean Hemmingson. So, the concept of chaperone is contrary to what we did in the Anfinson's experiment. The Anfinson experiment told us that the information regarding the tertiary structure of the protein resides within the primary structure of the protein, the amino acid sequence of the protein. But when we are talking about chaperones, we say that even though the information resides within the primary structure of the protein, yet many large proteins, many proteins which undergo a formation of a quaternary structure may require a helping hand or a chaperone, which is another type of protein which does not participate in the final structure, but binds to the early stages of protein structural and conformational journey. So, we had done the chaperones, and we have studied about molecular chaperones, which are the proteins which have a tendency to recognize all those hydrophobic residues that are exposed, and they inhibit aggregation. They prevent two misfolded molecules to come close together, that may prevent aggregation. And all these proteins mostly are heat shock, and they are expressed more during the thermal damage. So, they are induced when there is a high temperature, and at high temperature we know protein undergo lot of aggregation. So, this protein, which is the HSP, prevents and protects the proteins against denaturation. Then we have other chemical chaperones which are present. Many crowding agents also give the protein unfolded chain a push towards the native conformation, and we can use these and in vitro experiments as well. These crowding agents are glycerol, trehalose, trimethylene oxide and glycine, and various detergent and lipids as well. So, these we have discussed about chaeotropes and cosmotropes in our previous lecture under the Hofmeister series. So, you can pick some of the ions from there and think about them, which can prevent aggregation and promote native state formation. Also, there are some drugs which have been discovered that can rescue the molecules when they are going down the aggregation pathway. Many a times they can prevent degradation, and they can be proteins, they can be ligands and cofactors or competitive inhibitors, and they bind to specific conformation and stabilize native conformations and prevent misfolded proteins to aggregate. So, this was a reprise on the chaperone. Another machinery which is simultaneously working is, even though there may be misfolding and aggregation occurring within the cell at the same time, in an aged cell, a cell where there has been lot of stress, lot of environmental stress, oxidative stress, lot of reactive oxygen species present, that kind of a cell will have a tendency to have more and more aggregates of proteins. So, what happens to those aggregates of proteins? They are taken care by the protein quality control system or the protein homeostasis. We will discuss this system in detail, but I want to point out here is that there is a check during the folding of the protein, and there is a check, a quality control check, even after a protein has aggregated or has formed a misfolded insoluble fibril. Many a times these mechanisms are able to simultaneously balance out and clear out the degraded and the disease causing amyloid fibrils and aggregates and convert them back to the amino acids. So, we will discuss these systems. So, now let us talk about misfolding and aggregation. So, if a protein acquires a sufficient number of persistent non-native interactions such that it significantly affects its overall architecture and biological function, then we call it is a misfolded protein. These misfolded proteins will show aggregation, and aggregation by autocatalytic mechanism can form amyloids and further fibrils. So, mostly you will find how it progresses, that there is a core fibril structure which has all the hydrogen bondings involved in the peptide backbone, and the tendency of this fibril varies drastically between sequences. So, there are some sequences which tend to form more aggregates and some sequences which will not easily form aggregates. Then there is a concentration dependence, as we have discussed. Then nucleation rates may depend on the protein itself. Many mutations can modify the primary structure so much so that the protein never folds properly and it misfolds and aggregates. And there is a destabilization of the native folded protein by changes in the environment of the protein, its pH and other mutations, and it may expose the precursor leading to further fibril formation. So, the critical thing you will realize is that aggregation prone proteins will have lot of hydrophobic amino acid residues. They will have a predisposition for beta sheet formation, and they will have a low net charge. All these things will suggest that the protein will go towards a aggregation pathway. Many a times when I am saying beta sheet predisposition, many a times if there is a aromatic residue within the beta sheet, it further causes the beta sheets to become aggregated, and fibril formation occurs very quickly in those. So, there can be various alternatives concerning how the precursor is going to be formed from the native protein. There could be incomplete proteolysis of the protein, as you will see in the case of Alzheimer's disease, and that causes a misfolded component to be released, which eventually cause fibril formation and causes rapid aggregation. Sometimes protein aggregation can result in different intermediate forms. You will not always have a fibril, but there are subsequent oligomeric forms, protofibril forms, which will give rise to a large fibril. What are the triggers of misfolding? As I was talking about, that there are certain environmental, physiological stress that are involved in aging of a cell. Any cell which is newly formed and does not carries all this stress, does not have any mutation, will be having all the machineries working for protein degradation to take care of all the junk protein that is formed. But in case of an aging cell, you will encounter that there are a lot of reactive oxygen species formed, and those reactive oxygen species will further cause chemical changes into the protein structure and cause the protein to get destabilized. There can be several post-translational modifications like glycations which will cause the protein to further go towards the aggregation pathway. So, this is another diagram which talks about how quickly a protein is synthesized, then it is in unfolded state. It can go either ways, it can go towards a disordered aggregate state, or it can be degraded, recognized and degraded by the two machineries that I had discussed. It can form an intermediate, and this intermediate state can either again has a tendency to form aggregates, or it may form prefibrillary species. So, accumulation of further of these prefibrillary species will result in amyloid fibril formation, and these amyloid fibril formation, if everything is going all right, will be taken care by the protein quality control or the protein homeostasis machinery, and will be degraded. Again you will have recycling of the amino acids, but if everything is fine, the native protein may form its normal fibril or normal globular protein structure. So, we stop this discussion at this point where we have understood what is the important aspect in the protein structure itself that causes it to misfold, what is the difference between the thermodynamic stability of a misfolded and a normally folded protein, and we have also seen how amyloids are formed by accumulation of beta structure within the polypeptide. Students, in this section we are going to talk about what is the mechanism of amyloid formation in the misfolded and aggregated proteins, and what are the diseases that can be caused by this misfolding and aggregation. So, what is the mechanism of amyloid fibril formation? In this mechanism, the three important steps are: first, the alignment of the molecule to form beta sheets, largely depending on hydrogen bond formation between two separate chain of polypeptide. So, you do not have just one chain, you have two chains that are going to interact here. Then there is a cross beta structure, as you can see in the diagram here. This is a cross beta unit, and this involves van der Waals forces, and there is a steric zipper like structure formed between the two polypeptide chains, and eventually this cross beta structure further forms the fibril, and further non-covalent bonds are formed, and as you can see, a very stable insoluble aggregate is formed. Here also you can see that there is a soluble aggregate which is having lot of beta strand structure in the shown in the orange, and this beta strand structure will stack perpendicular to the axis of the strand, and further more and more strands will interact and will give rise to a protofilament, and all these protofilaments will give rise to a mature filament. The common feature of all protein conformational diseases is formation of an aggregate which is formed by destabilization of an alpha helical structure. So, when we will have an alpha helix structure converted to a conformation when there is more of beta sheet, and these beta sheet are formed between alternating peptide strands. There is a linkage between these strands caused by hydrogen bonding, and this gives rise to a aligned pleated structure and gives more stability as compared to a native beta strand we have seen. Such beta linkages with a pleated strand from one molecule being inserted into a pleated sheet of the next leads to a hydrogen bond formation between the molecules. So, there is a prerequisite that there has to be one donor peptide sequence and one beta sheet which is an acceptor of the other peptide sequence. See here in this diagram, you have entirely an alpha helical protein which undergoes such a conformational change caused by either chemical changes, environmental stress, oxidative stress or mutation in its primary structure, that it results into a beta sheet, and this beta sheet further causes more and more of the alpha helical structure to become beta sheet, and this will tend to form beta sheet like aggregates and fibrils. And the fibrils do not stop in that, there can be elongation of the fibril where you have prefibril oligomers which are soluble aggregates, they form protofibrils which are soluble aggregates, and they may form mature fibrils which are toxic, and they can promote secondary nucleation of even other proteins to form aggregates with them. This is also what you will encounter when we will see Alzheimer's disease. So, this is the amyloid formation which we encounter in the case of Alzheimer's disease or in case of Parkinson's disease. So, there is a native molecule, it will interact with its beta sheet structure in a molten globule, there is loss of tertiary structure, but some of the secondary structure is maintained. It can either be denatured or this molten globule can further undergo the misassembly pathway, the aggregation pathway, and give rise to self-associated prefibrillar structures, and eventually it will form the fibrils that we talked about. Another important aspect of misfolding is when we talk about cancers. Cancers also have a mechanism in which there is loss of function of the gene which is responsible for tumor suppression. So, the gene we are talking about here is the p53 gene which encodes for the tumor suppression protein, which are the transcription factors that regulate the cell cycle and apoptosis. This protein which is derived from the gene p53, it is also amyloidogenic, and we can see it can form amyloids, the structures that we have just seen. So, when there is here also you will encounter two type of toxicity. One toxicity is that because of the mutation the protein had lost its function. So, there was no longer it was able to inhibit the tumor, it was no longer able to suppress the tumor, and again because it has lost this function and it gained a function of being amyloidogenic, it will form long fibrils. So, wild type p53 loses its anti-cancer activity upon aggregation, while the mutant proteins show enhanced amyloidogenicity which shows accelerated aggregation. So, it will cause even autocatalytically more and more aggregation, and 10 types of cancers can be cited as examples for this type of misfolding of this protein to result in the disease. We can summarize protein misfolding diseases into familial, sporadic and transmissible, and we will see the examples of all these. When you have some of the misfolding diseases are genetically inherent, where there is a mutation in the protein primary structure such that it never attains its native conformation, and the symptoms of these genetically acquired diseases will be visible in childhood in young ages. Then there are sporadic, which may occur without any genetic cause at all. There can be environmental due to pesticides, heavy metals are implicated in the cases of Alzheimer's and Parkinson's. So, due to some aging related stress accumulation, incorrect lifestyle and chemicals in the environment, there can be sporadic emergence of protein misfolding diseases. Then there are certain transmissible protein misfolding diseases as well, and we will see the example of prions which cause spongiform encephalopathies. So, you have these diseases and there are the proteins that are responsible for it. This list can be helpful for you to remember what disease is caused by the responsible protein, the aggregation or maybe loss of function or loss of proper trafficking of the particular protein. So, it is not indicating the function which is lost, but this table is indicating you the diseases and the proteins that are responsible for this. So, first let us focus, when we think about prion disease, we come to think of all the diseases which are causing neurological diseases. We think of Alzheimer's and Parkinson's or prion diseases or mad cow disease, all these are pertaining to the brain. But it is not so that all misfolding diseases are going to be pertaining to the brain. Some of the non-neurological aspects of protein aggregation and diseases can be seen in the peripheral tissue. There is going to be accumulation of an amyloidogenic gene product, and some of the type 2 diabetes even are now described having protein misfolding and aggregation as the basis of disease of adult onset diabetes. Then there are cataracts, atherosclerosis, hemodialysis related disorders. In this disorder, there is no filtering of a particular protein which accumulates in the body and causes aggregation and formation of amyloids. And then several proteins such as fibrinogen, lysozyme, transerythrin, all of these can result in misfolded aggregates which give rise to amyloids. So, what is amyloidosis? Amyloidosis is going to be of two types. It can be systemic or it can be organ limited. We will see the example of organ limited as in case of diabetes. And then there is formation of amyloid fibril shown in the picture here. The amyloid fibrils are not just one type of fibril, they can give rise to plaques, they can give rise to Lewy bodies or annular pores which causes breakdown of the cell membrane and death of the cell in which they are formed. The basis of many of the neurological diseases are the formation of these plaques and pores. There can be several alterations which occur in the protein that can cause it to give rise to all those four important processes that were occurring, the mistrafficking, mislocation, improper folding, so and gain of toxic function. So, there are diseases which we have characterized on the basis of what alteration occurs in the protein. So, there is an altered protein structure, there is misfolding, and misfolding promotes enhanced aggregation. You will find it in the Alzheimer's, Parkinson's, prion diseases, and then diabetes and hemodialysis related amyloidosis. Then certain mutations can cause misfolding and again they will cause aggregation. So, familial amyloid polyneuropathy, systemic non-systemic neuropathic amyloidosis and cases of familial AD and PD. So, there are cases of Alzheimer's and Parkinson's which is early onset and can be genetic as well. The altered protein structure which results in impaired function of the protein, very important protein here is superoxide dismutase 1, important protein is our tumor suppressor protein. So, if these proteins lose their function, then they are not able to perform a very critical function. In case of SOD, it is taking care of all, scavenging all the reactive oxygen species and preventing oxidative damage to the cell, and if this protein is not going to work, that means the cell is going to accumulate more and more oxidative damage. We will discuss what then oxidative damage does to the cell and the protein structure. Then there can be changes in the supramolecular structure. Many a times proteins form quaternary structures, and if these quaternary structures are not properly formed, then they may also result in loss of function. Important example we will discuss is sickle cell anemia and cataracts. There can be altered protein structure because there is change, there are mutations. We will see polyglutamine disease, which is Huntington's disease, and then there are chemical changes which occurs post translationally, which can cause the or promote the protein to become misfolded. A very simple example, we have been learning this example since our childhood, is about sickle cell anemia and how this also is related to protein misfolding. So, you have the sickle cell shape of RBCs and normal red blood cells compared here, and the difference here is of just one amino acid, which is one glutamate changed with one valine, and this 1 point mutation is enough to change the structure of the hemoglobin. So, you can see in the diagram, this is the normal hemoglobin and here it is the sickle cell hemoglobin structure. The sickle cell hemoglobin structure, this protein has a tendency upon oxidative stress, when there is deoxygenated environment, lack of oxygen in the tissues, what will happen? It will cause clumping of the hemoglobin, and this will cause exposing of the hydrophobic patches. See the yellow patches getting exposed, and these lead to the polymerization of the sickle cell hemoglobin. Once this polymerizes, it has a lower capacity to carry oxygen. Also, it may change the elasticity of the red blood cells, causing it to break, causing it to form the sickle cell, and there is extensive tissue damage and there is anemia. And all these sickle cell hemoglobin molecules will form strands of 7 hemoglobin molecules together, and they will bundle to form a 14 strand helix, and these pack extremely densely and they cause eventually the red blood cells to rupture. So, this is a single point mutation which is causing loss of conformation leading to aggregation. Then we will talk about some systemic amyloidosis which are non-neurological. Here you will have the primary systemic amyloidosis, where it is caused by disposition of fibrils consisting of misfolded immunoglobulin light chains or fragments of light chains derived from proteolytic degradation. If there is improper proteolytic degradation, the product of the immunoglobulins will cause aggregates to form. So, the onset of primary amyloidosis is around 65 years, and there is accumulation of these amyloid structures around the heart and the kidney. Many a times a secondary systemic amyloidosis can also occur in patients which are suffering from chronic infections, diseases and cancer. There can be a protein which is responsible, the serum amyloid A protein, which is released. In case of an inflammation, there are lot of inflammatory cytokines that are released, and these cause the liver to produce this serum albumin, serum amyloid A protein, and this serum amyloid A protein accumulates around the kidneys, around the essential spleen, liver and kidney organ, and causes the disease. So, many of the proteins can form these kind of amyloidosis. This could be transerythrin, lysozyme, fibrinogen A, apolipoprotein A, and A1 and A2. Similarly, the case is of type 2 diabetes. As we were discussing, diabetes is also, this case is the case of adult onset diabetes. Here there is accumulation of a protein amylin, which is the isolate amyloid polypeptide IAPP, a 37 amino acid long peptide, and this peptide, once it is mutated or because of environmental changes, bad lifestyle, can cause aggregation around the islets of Langerhans in the pancreatic cells. So, the pancreatic islet B cells, which are the cells responsible for producing insulin, get engulfed by this aggregate or amyloid formation, and subsequently there is loss of the pancreatic islet B cells, which will cause further decrease in insulin and which will further cause glucose homeostasis to be disrupted. So, you can see the structure of a human amylin, and these are the regions which are responsible for further aggregation, and aggregates can formed around the pancreatic islet B cells. Then another case is of cataracts. Cataracts occur in the eye, and many a times cataracts can be familial as well as some of the cataracts occur with age. With age, again what happens, there is accumulation of lot of oxidative damage and cell stress in the cell, and it is not able to take care of it. It is not able to incorporate the machineries responsible for taking care of all the junk, misfolded, not useful proteins. In this case, I would like to say alpha crystallin itself is a kind of chaperone, and imagine if a chaperone itself gets mutated, what will happen in the eye? It will not be able to take care of all the junk, and more accumulation of aggregates will occur. So, there are two genes which are responsible, CRYA and B genes, which give rise to the crystallin A and crystallin B. So, these are small heat shock proteins. When they are present, they occur as heat shock proteins, taking care of all the proteins that are present in the eye. They maintain the refractive index of the eye, which is very important for clear vision, and they maintain the microfibril stability. I would like to say that all the proteins that are present in the eye lens are first formed even during your embryonic stage, and what the cells were present during the embryo state will be there with you till you are 70 and more. So, there is a long life of these microfilaments that are present, and the chances of accumulation of aggregates and misfolding will enhance if the chaperone system itself does not work properly. So, you can see here is that we are showing a truncated version of a protein occurs. There is partial unfolding, and the crystalline chaperone forms substrates which are soluble, and then it may form aggregates which further cause clouding of the lens, and it will distort the vision. Another disease very famously which we recognize by the scientist Sir Stephen Hawking is amyotrophic lateral sclerosis, and these days if you remember, we had the ice bucket challenge also to bring awareness to this disease. This is also a disease which is affecting the nerves, and here there is loss of function of the SOD1, which is the superoxide dismutase protein, which checks the oxidative stress, which checks and scavenges the reactive oxygen species from the cell, and there are and this is a protein which has very critical ion binding centers. As you can see from the diagram here, there can be copper or zinc binding sites. Superoxide dismutase is a dimeric molecule, so each of them will have a ion binding site. Upon mutations in this protein, there is going to be improper binding to the metal. If there is improper binding to the metal, it is not able to perform its function of scavenging the free radicals, and also it will result in formation of amyloids. So, there is loss of function, and there is death of the nerve cells, and when the death of the nerve cells occurs, eventually the muscles also get atrophied because there is no longer any message coming from the nerves and the muscles lose their function. So, this is the structure of superoxide dismutase in ALS. Another important aspect is improper degradation, that is when there is failure of correct degradation of the junk or wasted protein, when the ubiquitin and proteasome system is unable to decipher if it is the correct protein that they are degrading or it is a unfolded junk protein that they are degrading. This is encountered in cystic fibrosis, and the protein responsible for this disease is cystic fibrosis transmembrane conductance regulator. This protein, as you can see in the diagram, is a chloride ion channel. So, when it is a chloride ion channel, it maintains all the epithelial cells, all the exocrine functions of cells are maintained by this channel, and this results in formation of a very sticky mucus plaque on the surface of the cell, such that there is impaired oxygen exchange in the lungs. It is also affecting the GI tract wherever you encounter these cells. So, in this mutation there is a deletion of a single phenylalanine residue at the position 508 in the protein CFTR. This mutation interferes with the biosynthetic folding of the cystic fibrosis transmembrane conductance regulator, and it pushes it towards degradation, and it is not expressed at the plasma membrane of the cell. So, it is absent from this ion channel gets absent from the plasma membrane, it is not able to perform its function. Also, whatever it is formed gets diverted to the proteasome machinery for degradation. So, there is no protein present for ion maintenance, and the sweat and intestinal secretions and secretions in the lungs become highly sticky and mucus like very sticky mucus and rubbery. Then there is again a case of improper localization which we encounter with the alpha 1 antitrypsin, which is a protease inhibitor. A protease inhibitor is an enzyme which will prevent proteases to become activated at all times. Imagine if all the proteases are going to be activated, then they will cause damage to the vicinity, to their environment, to the organelles they are present. So, you need to have a check on when the protease get activated. I would like to remind you, proteases are enzymes which break down proteins, and they are not present as active molecules at all times. So, there is always a precursor molecule which is released, which has to be further cleaved and made active. But in case of protease inhibitor, there is no check on proteases, and they will cause hydrolysis of any protein that they encounter. So, in this case also, if this protein is not present, then there is no inhibition of the proteases, and this also affects the lungs and connective tissues. In this the antitrypsin, the mutant of this protein, fails to completely properly fold, and it is only retained in the ER, and this misfolded protein is not degraded. So, there is accumulation of the mutant in the liver cells. Then there are some mutations, and we are encountering here the mutation which occurs in keratin. This causes a disease of several lesions that can be formed in the skin, and it occurs because keratin is unable to form the correct alpha helical structure, but it forms long intermediate filaments, and in this the protein is misfolded and leads to, and whenever there is whenever there is mechanical stress, it will cause misfolding. In this case, you do not need both the protein both the alleles to be mutant, even one mutant allele is sufficient. So, even a heterozygous individual will make both a mutant and a wild type, and the mutant version will be sufficient to cause misfolded and will compromise the function of keratin in all the epidermal cells. Then we have several amyloid fibril deposits, we will just summarize it here, which are going to be incorporated in the neurodegenerative diseases. So, you have Alzheimer's disease, it was discovered in 1908 by Alois Alzheimer. This is the most commonest, most prevalent neurodegenerative disease, and it has a massive loss of neurons. So, you can see this is a healthy microtubule which is present in the neuron and gives structural stability to the neuron, but in case of Alzheimer's there is no structural stability and there is tangles which are formed. So, we call them neurofibrillar tangles. Two proteins are important in Alzheimer's disease. One is the amyloid A beta protein, which forms extracellular plaques, and then you have the tau protein which forms intracellular NFTs or neurofibrillar tangles. Alzheimer's, as you know, has memory impairment and inability to think by themselves. Generally it occurs by aging, but it can also be familial and also be can also be induced by environmental stress, pesticides and metals. So, what is the mechanism of amyloid formation here? So, the protein responsible here is A beta peptide, which is in the transmembrane region. So, this is the entire protein. As you can see, the protein has a calcium binding region, it has a transmembrane region. So, two enzymes can act on it, which are alpha secretase and beta secretase. In normal condition, alpha secretase acts on it, chops it, and further gamma secretase also chops it, and none of the structures formed here are amyloidogenic. All these are non-amyloidogenic, that is, they do not cause fibril formation. But if there is beta secretase involved here, which causes chopping of the protein at the A beta peptide region, then this A beta peptide will form further aggregates, and these aggregates will further oligomerize and cause plaques and fibrils. So, you have improper folding occurring because there is improper chopping of the peptide. This also is important, we will discuss it when we talk about proteases will chop proteins into those components or fragments that can further cause misfolding. And another disease here similarly is Parkinson disease, but the protein responsible here is alpha synuclein. This disease was discovered by James Parkinson, and it is the second most important neurodegenerative disease, and it is also genetic and as well as environmental and age related. So, you can see the component here form is a Lewy body, protein responsible here is alpha synuclein, and Parkinson's disease causes these Lewy bodies to form, and they will prevent the correct form the dopaminergic neurons to function properly. When the dopaminergic neurons do not function properly, they do not secrete enough amount of dopamine to cause muscle stimulus, and hence there is motor nerve dystrophy in this kind of neurodegenerative disease. As you can see, there is a diagram of accumulation of synuclein protein. So, we have seen that there are neurodegenerative as well as non-neurodegenerative diseases that can be caused by misfolding and aggregation of protein. We also saw cancers can be caused by aggregation and misfunctioning of the protein. So, we will further take up other examples of diseases which result from misfolding of disease of the proteins. Thank you.
On that note, I would like to thank ma'am for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.